Hello, everyone. This is Dr. Muzamil Liaqat Ali, Assistant Professor of Biochemistry. Welcome to lecture number 1 of our Lipid Biochemistry series. Lipids are a highly diverse group of biomolecules that play critical roles in energy storage, cell membrane structure, signaling, hormone synthesis, and many disease processes, including the atherosclerosis, obesity, and inflammation. So mastering lipid chemistry today will form the strong foundation for the understanding of the lipid digestion, beta oxidation, cholesterol metabolism, eicosanoids, and the upcoming lecture series, right? So let's begin with clarity, depth, and strong clinical correlation. So in this series, we will cover classification of the lipids and their general biological functions, fatty acids, their nomenclature, classification, properties, essential fatty acids, omega-3 and omega-6, and trans fats, physical and chemical properties of fats and oils. We'll discuss in detail the saponification number, iodine number, acid number, and rancidity. And furthermore, complex lipids like the phospholipids, glycolipids, and gangliosides. Along with finally, we'll conclude this series on the cholesterol, bile acids, and lipid peroxidation. So the learning resources you'll have, you can cover these topics from the Lippincott, Harbour, and Chatterjee, and of course the lecture notes. So at the end of this lecture, we will be able to understand or define the lipids, explain their unique solubility properties, classify lipids into simple, complex, and derived types of lipids with examples, describe the structure and biological significance of the waxes, triglycerides, phospholipids, and glycolipids, along with sterols. And finally, discuss the general biological functions and biomedical importance of lipids. So let's start with the definition of lipids. Lipids are the heterogeneous group of organic biomolecules that are fatty acids or their derivatives. So what about their key characteristic features, right? So they are insoluble in water, but readily soluble in non-polar organic solvents such as chloroform, ether, and benzene. They contain carbon, hydrogen, and oxygen, sometimes nitrogen and phosphorus as well. More reduced than carbohydrates, therefore yielding 9 kilocalories per gram energy. That is 2.25 times more than carbohydrates or proteins. So this hydrophobicity is the basis of their role in cell membrane and energy storage. So now let's classify the lipids. Lipids are broadly classified into 3 major groups: the simple lipids, complex lipids, and derived lipids. The simple lipids, these are the esters of fatty acids with alcohol. So there is no additional group is present. That includes the waxes and the triglycerides. The triglycerides include fats and oils. What about the complex lipids? They contain additional chemical groups, the phosphate and the carbohydrate, or etc. The examples are the phospholipids, glycolipids, and sulfolipids. So what about the third type? That is derived lipids, obtained from the hydrolysis of simple and complex lipids. The fatty acids, steroids, derived from the cholesterol molecule, eicosanoids, and the terpenes. These are the examples of simple derived lipids. So as you can see on your screen, this shows the classification of the complex lipids, right? The family of the lipids, right? So let's move on to simple lipids, the waxes. So simple lipids are fatty acid esters of different alcohols and carries no other substance. These lipids belong to a heterogeneous class of predominantly non-polar compounds. And they are mostly insoluble in water, but soluble in non-polar organic solvents such as chloroform and benzenes. So simple lipids can be further classified into waxes and triglycerides. So waxes are esters of long-chain fatty acids with high molecular weight alcohols that ranges from the 12 to 32 carbon atoms. That they function as they provide a waterproof coating on plant leaves, fruits, and insect cuticles. Forms protective layer on bird feathers and the mammalian skin or the air wax. So waxes act as an insulator or prevent excessive water loss. So ear wax, cerumen, is a type of wax that protects your ear canal. Now let's move to the simple lipid, the second type, that is triglycerides. So simple lipids can be further classified into the monoacyl glycerols or the monoglycerides, the diacyl glycerols that contains two molecules, right? So diglycerides found in the plant leaves, the one fatty acid is replaced by the sugar that is galactose. And the third one is the triacyl glycerol or the triglycerides. Triglycerides found in the seeds and animal adipose tissue. And this is the lipid storage form in humans. So where in the body this pretty molecule, the triglycerides or the simple lipids, get stored? The answer would be very simple, the adipocytes. So as we know, the structure of the triglyceride that contains one molecule of glycerol esterified with the three fatty acids. So we can further classify this pretty molecule into fats and oils. So fats are solid at room temperature. That is more saturated form, right? So solid at room temperature, more saturated fatty acids are present in its structure. Oils, liquid at temperature or the room temperature, so they got more unsaturated fatty acids. So they are stored in adipocytes and serve as a body's concentrated energy reserve. So what about the variation in the triglyceride structures? Fatty acids composition vary according to biological needs. Membrane lipids require fluidity, so more unsaturated fatty acids are required to provide the fluidity in a lipid membrane. So animals in cold climates have more unsaturated fats. Butterfat remains relatively fluid despite being mostly saturated due to short-chain fatty acids. As you can see on the screen, the pretty molecule of the triglyceride contains the one molecule of the glycerol and three fatty acids are esterified to form a complex molecule that is triglyceride. So glycerol molecule provides the backbone and the two fatty acids are present along with it, right? And there may be a phosphate group, nitrogen base, the choline, ethanolamine, or serine, or inositol may be present to produce a more complex structure or more complex molecule. So major examples are the lecithins, cephalins, and phosphatidylinositol molecule. That is pretty important in the signaling or cell signal transcription, right? So they are amphipathic in nature and they have both hydrophilic and hydrophobic parts, enabling them to form a lipid bilayer or the bilayers. Now let's move to the complex lipids, the glycolipids. Glycolipids, basically complex lipids, there are two primary types. The phospholipids that form the core structure in the glycerol. The core is provided by the glycerol molecule, and this phospholipid, the part of the cell membrane, the chylomicron, the lipid, the lipoproteins, right? And its structure, the two fatty acids are present, but the third fatty acid is being replaced by the phosphate group and amino alcohol. So phospholipids are of further two types, glycerophospholipids and sphingophospholipid. And the second type is the glycolipid. The core is the sphingosine or sphingol molecule, right? Along with that, a fatty acid and the sugar moiety is present. So this pretty molecule, the glycolipid, is a part of the sphingomyelin. That is of further, you can further classify that pretty molecule into two types, the neutral glycolipids and the acidic glycolipids. The neutral glycolipids include the cerebrosides and globosides. And the acidic glycolipids have further two molecules, the gangliosides and sulfatides. As you can see, this shows the molecular biology of the cell, right? A lipid bilayer structure. Phospholipid bilayer or the membrane that shows the hydrophilic, the outer part of the membrane, and the hydrophobic part, the inner part. That shows the fatty acid molecules and the glycerol molecule. And the polar head, that is, hydrophobic head, right? That is present outside the, that is interacting with the phospholipid membrane. And the inner, that is hydrophobic in nature, that is present inside or buried inside. So complex lipid, the phospholipids, resemble triglycerides in structure, except one of the fatty acid is replaced by the compound containing a phosphate group or occasionally by the nitrogen. So most prevalent form of the complex lipid, that is phospholipid, is lecithin. So examples are the phosphatidylcholine, phosphatidylethanolamine, or the phosphatidylinositols, or phosphatidylserine, or the plasmalogens, right? So let's talk about the phospholipids. The significant use in the feed industry as emulsifiers. This is one of the use of the phospholipids. The lipids form emulsion in water. And its basic structure is the backbone is provided by the glycerol, and the two fatty acids along with the phosphate molecule and amino alcohol is present in its structure. Phospholipid sources are the liver, egg yolk, soybean, wheat germ, and the peanuts. What about the second class of the complex lipid, that is the glycolipid? The carbohydrate component is present in its structure. The core structure is provided by the sphingosine, or the backbone is provided by the sphingosine or sphingol molecule. And along with that sphingosine, the fatty acid and the sugar moiety is present. That is of further two types, the neutral and the acidic glycolipids. The neutral glycolipids include the cerebrosides and globosides, and acidic glycolipids include the gangliosides and the sulfatides. So what about the third class? That is the derived form of the lipids. The very first one is the prostaglandin, synthesized from the arachidonic acid, and they are of very important in their functions. They provide several metabolic functions. So derived lipids also include the steroids, derived from the cholesterol, the ergosterol, and the bile acids are the examples. The terpenes made by the plants, also present in the form of the carotenoids or xanthophylls in plants. So what about the sterol? Sterol is basically a 3-cyclohexane and 1-cyclopentane used together to form a sterol structure. Compounds with the multi-ring structure, that is, you can label it as a sterol, that is insoluble in water, present in plants and animal foods both. So major sterol is cholesterol. However, cholesterol is found only in plant products manufactured in liver, and the high content in organ meats and animal yolk, that is the hallmark feature of the sterol, that contains 3-cyclohexane and 1-cyclopentane fused together to form a multi-ring structure, that is sterol. So what about the common sterol compounds that are present in your body, right? Or maybe found commonly. So very first one is the cholesterol, and the along with the vitamin D3, testosterone, and stigmasterol. That is the plant source, right? The phytosterol. So what about the common physical properties of lipids? Soluble in non-polar organs, solvents, they contain carbon, hydrogen, and oxygen. sometimes nitrogen and phosphorus as well. They include fats and oils, mostly triglycerides. The fats are the solid at the room temperature. What about the oils? They are liquid in temperature and they have polyunsaturated fatty acids present in its structure. So physical properties of the lipids also include they are highly or more reduced than the carbohydrates and provide 2.25 times more energy. So what about the functions and properties? Concentrated source of energy. They are, this is one of the hallmark of the lipids. They provide 9 kilocalories per gram. So they act as an energy reserve. They provide insulation to the body from the cold, maintain body temperature, mechanical insulation, protects vital organs. Along with that, it also acts as an electrical insulation, provide electrical insulation, protects the nerves, help conduct electrochemical impulses, myelin sheath. This is one of the pretty phenomena you can have in your books, right? So supply essential fatty acids, linoleic acid and linolenic acid, along with the formation of the plasma lipid membrane, right? So along with that, synthesis of prostaglandins from the fatty acids and transport of fat-soluble vitamins and palatability and the aroma that is being provided by the lipids. So thank you very much for your attention. If this lecture clarifies your concept, please like the video, share it with your pathmates, and subscribe to the channel. And don't forget to hit the notification bell so you never miss the upcoming lecture. Thank you very much. This is Dr. Muzamil Liaqat Ali signing off. See you in the next lecture. Thank you.